Okay, good morning, everyone. I'm happy and excited to welcome you to the NA3RC and IQ Microphysiological Systems webinar and workshop series. Today, our focus is going to be on kidney MPS. Before we get started, I'm going to read a brief disclaimer. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops is intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither NA3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. For a little bit of housekeeping, please try to submit questions via the new Q&A. We'll answer questions at the end of the webinar as time permits. If you're having difficulties finding the Q&A and you put your questions in the chat, I will still notice them. So a little bit of introduction. So who are these people putting on today's webinar? Well, my name is Dr. Megan LaFollette, and I am Program Director at the North American 3Rs Collaborative. We are a nonprofit that collaborates to advance science for the benefit of both people and animals. And we do this through facilitating 3Rs opportunities focused on refinement, reduction, and replacement. We work with partners across the field and across industries. So that includes academics, industry partners, biotech, government, other nonprofits, and more. This is a picture of just our leadership team and board of directors. We look for initiatives that we see have ample evidence, impact, and real-world practicality, of which, of course, we see microphysiological systems as a very promising replacement. And so that's why we seek to increase adoption and regulatory acceptance of MPS in coordination with animal studies. We currently have 46 institutions, 30 that are developers that are members of our group, and we are really excited for their support. We focus on 4 key efforts. That includes interfacing with end users via our MOU with the IQ, as well as just integrating outreach with other partners. We work to increase regulatory acceptance through presentations, publications, and talks with the FDA. We have created a technology expo, which I'll show in just a minute. And finally, we work for general education. Of this webinar is included in those efforts. So we do have presentations to introduce MPS to the more general new audience, as well as to accelerate regulatory acceptance. You can find both of those on our website. Of course, if you're a member of the IQ, hopefully you're at this webinar and workshop series. And we are working on 2 different publications as well. And then the one thing that I do want to highlight is our technology hub. It's a really wonderful resource. You can search by organ type currently, and we're going to be adding other filters and find all of the different commercial providers that provide those technologies. So just to introduce you a little bit to this series, so today we are going to be having a webinar about kidney MPS. These are typically fairly institutionally agnostic and are focused about general education about an MPS organ type. But we are also going to be having a workshop, which is where each company presents their own kidney MPS models, and they're really about specific commercially available MPS systems. The Kidney MPS workshop is going to be on November 9th, and we're going to hear from 6 different commercial providers of kidney MPS, as well as a Q&A. Today, I'm excited that we're going to be hearing from Dr. Jonathan Himmelfarb from University of Washington Medicine. I'm actually going to let Banu do her introduction before I read the bio. Awesome. Over to you, Banu.
Thank you. I can share my screen. Good morning, good afternoon, everyone. My name is Banu. I'm an investigator in GSK, but today I'm representing IQ MPS, the Strategic Partnership and Communications Workstream. Just a brief 5-slide intro about the IQ MPS for anyone new here. The current membership of the IQ-MPS is about 22 member companies, and each of these companies get about 2 individuals on the steering committee, and we do have several work streams that I will talk about in a minute. But overall, we have diverse representation from drug safety, 3Rs, ADME, and PKPD experts. And the leadership team, current leadership team, is Rhiannon Hardwick as the chair, and Anna Kopek as the vice chair, and Jason Eckert as the past chair. Overall, there are 4 goals of the IQ-MPS affiliate. The first one is to serve as a thought leader for both the MPS developers and stakeholder organizations in how to implement in an industry-wide scale and qualify the MPS models. The second goal is to provide a venue for cross-pharma collaboration and data sharing to facilitate industry implementation and, again, qualification of MPS. Third thing is create focused engagement between regulatory and industry bodies on the current status and evolving MPS field, again, with the context of an industrial setting. The last one, which is the group I belong to, is develop external partnerships, such as with the NA3RCs and collaborations to help enhance the inclusion of industry priorities. Amongst the 5 work streams, I'm going to, just in the interest of time, since we're 7 minutes past, I'm just going to say the one that's part of the IQ, which is presented here, is the strategic partnerships and communication, where we communicate industry perspective and we build relationships with key stakeholders to enhance the inclusion of the industry priorities. But there are other work streams, such as writing organotypic manuscripts, regulatory outreach, pilot project teams, and landscape gap surveys. For the external partnership timeline, from 2018, we started off with the NCATS Tissue Chip Consortia all the way to 2021. We do have an MOU, as Megan just said, with NA3RCs, engaged with Army and NIST, and we provide a year-end update and overview of landscape assessment gap surveys to the IQ Drug Safe Leadership Group. That's it from me. Very quick intro, just so we can get to the talk. Back to you, Megan.
Awesome. So I'm going to very quickly introduce our speaker today. Dr. Jonathan Himmelfarb is a board-certified physician and director of UW Medicine's Kidney Research Institute and the UW's Joseph W. Eschbach Endowed Chair in Kidney Research, president-elect of the American Society of Nephrology, and a UW professor of Medicine and Nephrology and adjunct professor of Medical Bioengineering. He will present a talk giving an overview and application of kidney MPS. So we are excited to turn him over today. Jonathan, I did mute you for just a minute. So if you want to go ahead and unmute and start sharing your screen.
Thanks, Megan, for the introduction and for the chance to speak today. I was asked to give kind of an overview of where the field of kidney on a chip is. And so I will try to do that in the next 45 minutes or so. And let's see if I can get the slides to move. That's always a challenge. Oh, good. So I'm going to start with an old slide, 10 plus years old now, from Margaret Hamburg, a quote when she was the commissioner of the FDA, that we are failing, very strong language from an FDA commissioner, to bring to patients the treatments that they want from us, and that we must invest in new methods, assays, standards, and models that will help speed the development, review, and approval of the products that our patients want. So this was strong language from the FDA at that time, and similar language from Francis Collins, who was the head of the NIH at that time. And kidney diseases, just since that's the focus today, are, I will say, a growing public health problem that affecting roughly 12 to 13% of the entire world's adult population, 10th leading cause of death in the US. And kidney failure is very challenging for patients with very short survival on dialysis, very expensive as well. And the challenge for us, as I am a nephrologist who still sees patients, is that we really have almost no cures for any kidney disease. We are getting better at treating kidney diseases, and I will say pharma has become increasingly interested in the kidney space, given the public health challenges, the number of people affected, and the success of the SGLT2 inhibitors for treating chronic kidney disease. So there is growing interest. We see it in both biotech and in pharma in finding new ways to treat and hopefully cure kidney diseases going forward. So why has the kidney been so refractory? I would say the kidney and the brain have been the organs in the human body most refractory to developing effective, safe and effective therapeutics over a number of decades, and both have been very frustrating areas. And it's probably because they're the most complicated organs in the human body. So the kidneys have a compartmentalized structure. Your two fist-sized organs get 700 liters of blood flow a day. Roughly 20 to 25% of the cardiac output goes to the kidneys. So they're the most vascular organs in the human body. And the structure, architecturally and functionally, is very complex. There's this compartment called the glomerulus here in the upper left. I don't know if you can see my arrow, which has got this intricate structure from this cell type called the podocyte. And basically, it's a bandpass filter that takes that 700 liters of blood, and on the other side of the filter is a protein-free filtrate that then goes down this incredible tubular structure here, the other compartment really of the kidneys, along with the interstitium, which consists of 60 or 70 different cell types, all of which sense the circulation, sense what the composition of the filtrate is, and have transporters that bidirectionally transport either reabsorb solutes or secrete solutes directly from the blood into the filtrate that becomes the urine. And the kidney has to maintain homeostasis for the whole body by keeping the blood in a constant state. So it's an architecturally complex organ, 60, 70 different cell types, functionally very complex, very much like the brain, and that's why it's been hard to make good therapeutics for kidney diseases. Well, just about a decade ago, because it was 2012, that for the first time ever, NIH partnered with DARPA and the FDA to come up with this idea of human organs on chips for precision medicine. The idea being, of course, that we can cure a lot of diseases in animal models that simply haven't translated into human treatments over time. And maybe if we work with human cells and mimic human organs and represent in chips the genetic diversity of humans, as opposed to most of our animal models where people use the same genetic background, we'll do a better job of converting promising agents into actual therapeutics. So this was the genesis of this program, and we competed in this program to make a kidney on a chip, thinking that if there was an organ system where microfluidics would be beneficial, where every cell, because of those 700 liters of blood and the 140 liter filtrate, of which 99% gets reabsorbed, every cell in the kidney touches fluidics constantly. So there are now a lot of different platforms out there 10 years later. We use the Nordis platform primarily simply because Nordis was a company spun out of the University of Washington, and we started working with them way back when in 2012. But there are many other, and if I'm missing, I apologize to anyone if I'm missing another commercialized kidney chip platform. But there are many of them out there. My own personal view is the platform is less important than the cell source and the cell types that you use, but the platforms all differ. Some are higher throughput, some are higher content. They have different architectures. Depending on your context of use, one may be preferred over another, but really all the platforms are probably, if you do your experiments well and you have a good cell source, you can probably get good data. But we use the Nordis system, which in this 70 nanoliter space here, again, I don't know if you can see my arrow, you can bioengineer cylindrical structures, and you can put two cylindrical structures side by side. So you can mimic a tubule and you can mimic a blood vessel in close apposition to each other. You can perfuse it luminally, you can collect the filtrate, and you can cross perfuse to mimic interstitial flow as well. In the newer generations of triplex chips here, you can do 3 experiments on 1 chip. So when we set out, we knew, and this was the first goal, I should say, of what we call Tissue Chips 1.0, the beginning of the NCATS effort, was focused primarily on building these systems and really focused on safety, nephrotoxicity in our case at the beginning. And the part of the kidney that's most vulnerable to nephrotoxicity, drug-induced kidney injury, is the proximal tubule. And this is a photomicrograph on the right of the cortex of a normal human kidney. And the cortex is 90+% proximal tubules and the surrounding vasculature, as you can see here. All the green are the proximal tubules, the blue and the red are the vasculature, and every tubule is surrounded by blood vessels in close opposition, and that allows the reabsorption of solutes and secretion of solutes. So rather than try to build 60 cell types into a chip, we said if we can build human proximal tubule cells and then peritubular microvasculature with microvascular endothelial cells and so-called pericytes, then we can mimic solute transport, and we can also assess for nephrotoxicity. And it took us a couple of years to be able to do this reliably and reproducibly, but after a while, with some trial and error on the conditions, 95% of the time we can bioengineer these beautiful proximal tubules. And as I'll show you in a minute, they really replicate the functionality, the physiology, and the response to injury of the human proximal tubule. And they maintain their epithelial phenotype when you put them in a three-dimensional architecture with fluidics for an extended period of time, as opposed to if you put human proximal tubule cells in a dish, two-dimensional dish, within 2-3 days they start to undergo epithelial mesenchymal transition, and they no longer really have the right cellular phenotype. So we learned to do this, and other groups learned to do this as well. And these were some of the, and in fact, the IQ Consortium, the MPS IQ Consortium way back when, put out a paper on requirements for a kidney on a chip. And basically, we could replicate all of the required functions and the response to physiologic stimuli in our proximal tubule on a chip. And of course, we're not the only people who could do this. And Jennifer Lewis, who I know is working with some pharma at this point in time, groups. She's in Boston at the Weiss Institute and the Beth Israel. And she's a wonderful 3D printer. And the proximal tubule actually has a convoluted shape. And rather than just having a cylinder, she prints the convolutions. And this is just one of her early papers showing, again, that these cells maintain fidelity with fluidics to what you would see in vivo. There's an apical, a bit of a brush border here, tight junctions, and cell polarity as well. So, you know, her group continues to perfect in different models, and you know, her work is really tremendous as well. This is one of her newer papers, and it just shows the degree of both transporter expression and polarity that she can get in a chip model as well. I should say we also we've demonstrated polarity and transporter functionality as well. So these proximal tubules now on chips that many people have developed really, really closely mimic what you expect to see in vivo. And here's a third group that I would consider leaders in the field. Roos Mazeroux in Utrecht in the Netherlands has done a lot of great chip work as well, also focusing on proximal tubules, and also now has come up with a way to incorporate the convolutions as well as a genetic model of cystinosis. So multiple academic groups keep pushing the envelope and incorporating new concepts into their work, and industry has adopted a lot of the commercially available platforms, and in some cases is working directly with some of the academic leaders as well. So once we're able to show physiology, the whole goal was response to injury. This was sort of the overall model for what we do in chips, that there are different cell types that can be injured in different ways by different initiators and mediators, but it's really the response to injury that determines whether you're going to have disease or not. And if you undergo a response to injury that's pro-inflammatory, pro-fibrotic, you're likely to end up with what we'll call chronic kidney disease. But you can also undergo reparative pathways and regenerative pathways. And really, it's not necessarily just the injury, but it's the response to injury that determines whether or not you're really going to end up with disease. So the first thing we've set out to do is say, can we come up with reliable markers, either cell-associated or in the effluent, biomarkers in the effluent for injury and for the different kinds of response to injury? And sure enough, we found that cell-associated hemoxygenase 1 was induced in the proximal tubules with almost every nephrotoxin that we could test. So it's a very reliable marker of injury in this proximal tubule on a chip. We also found collecting the effluent, which would be the equivalent of the filtrate that would turn into the urine in a human being, that, for example, KIM1, kidney injury molecule 1, is upregulated in response to almost every nephrotoxin as well. So we had very reliable injury biomarkers in our proximal tubule on a chip. And then we wanted to develop markers of fibrosis, and so we did that as well that could be used in chip. And I didn't put the slide in, but we had a third paper where we showed some regenerative biomarkers that could be assayed in chip as well. So then we had a toolbox. This took a while, but then we had a toolbox that we could use to start modeling injury and response to injury. And I'll take you through one story that shows you some of the power of chip work. There are other stories, but I'll focus on this one. It's called aristolochic acid nephrotoxicity, or nephropathy. So it turns out that about 30 years ago in Belgium, a little over 100 women, young women who were in a weight loss program, within a period of months, developed irreversible kidney failure, and many of the women went on to develop lower urinary tract malignancies as well. Previously healthy, 6 months later on dialysis and some developing malignancies. So this obviously caught people's attention. There was a CDC-like investigation, and it turned out there had been a mix-up in a Chinese herb preparation. One herb was substituted for another so that they were getting high doses of something called aristolochic acid. And this was published in the New England Journal back 25 years ago, in Lancet. The whole story was told. And there had been an entity that was very mysterious, but had been known for 100 years, called Balkan endemic nephropathy. And it had been known that there were villages along the Danube River in the Balkans where there were high rates of kidney disease in the villagers. And people were searching, what was the cause? They looked for lead, they looked for all kinds of things. And there was no consensus on this. Well, it turned out after the Chinese herb nephropathy story, people went back and they found that in the villages along the river, the Danube River, the aristolochia, the plant, was growing, and the seeds were getting mixed with the wheat that the villagers were growing. So whereas the women in Belgium got very high, like a bolus of aristolochic acid, high concentration, short period of time, in the Balkans, people were getting chronic low-dose exposure, but were developing kidney disease over time. And it turns out that aristolochic acid is now considered a class 1 carcinogen in the US. You can still buy it on the internet, even though it's banned in the US. And about 100 million people worldwide are still exposed to it, especially in Asia. Taiwan has the highest rates of exposure and the highest rates of kidney failure of any place in the world. And it's now known to be mutagenic by adducting to DNA and to proteins. And there have been 25 years of animal model research on this, but there was a lot of controversy from animal models about how aristolochic acid, after it's ingested, is bioactivated in the liver and how it becomes nephrotoxic. So one of our enterprising grad students took our kidney on a chip, put human hepatocytes in another chip to be a liver on a chip, mixed liver and kidney media 1-to-1, and infused aristolochic acid to see if she could mimic what's seen in terms of bioactivation of aristolochic acid to make it nephrotoxic. And the readout can be very simple, and that's an important point to make. You know, you don't always need RNA-seq readouts. She did live dead staining. Here's aristolochic acid here. She perfused it just through the kidney, 10 or 25 micromolar, and Green is live, red is dead, and there's really no toxicity, maybe a few dead cells at 25 micromolar. But if she pre-perfused it through the hepatocytes, you start to see dead cells at 10 micromolar, and the entire tubule is necrosed at 25 micromolar. So she could show you need to bioactivate, the liver needs to bioactivate aristolochic acid to make it nephrotoxic. She then did a more sophisticated experiment with antibodies to the aristolochic acid DNA adducts and showed that even at 10 micromolar, these adducts are forming only if you pre-perfuse through the liver. So the metabolic pathways of aristolochic acid are complex, and that's part of why there was a lot of arguments from the animal models about what was the toxic agent. There's cytochrome P450s in the liver, oxidases that are thought to be a detoxification pathway, whereas reductases are thought to be activating, form the N-hydroxide, but that can also, there are sulfur transferases in the liver that can sulfate it, and it wasn't clear exactly which was the etiologic agent. So she reasoned that whatever was the toxic agent wouldn't need to be pre-activated by the liver. It would be equally toxic if you just perfused it through the kidney chip. So she first perfused the N-hydroxide through and everything stays green, so that's not the toxic agent. She then put the sulfated form through and replicated what would happen as if you pre-activated it by infusion through the liver. And that made a lot of sense to us because sulfated compounds are anionic and they're targets for secretion in the proximal tubule through organic anionic transporters. And if that's the case, we could block that with an agent, probenecid, that's used to block oats, so-called oats on the basolateral surface. And she showed, yes, if you pre-treated with probenecid, it was protective against the toxic agent. So this one graduate student on her own in 3 months worked out the entire pathway after 20 years of animal model research, whereby the sulfated form is then exported, she showed, out of MRP3/4 out of the liver, it goes to the oats and the kidneys, it's electrophilic, it binds DNA, and it destroys the proximal tubule. So it gives you a sense of the power of some of these in vitro systems in ways that animal models can't really give you the answer necessarily. Oh, I put this in too. So here's another kind of cool example, polymyxin B. So I'll focus first on safety, then I'll talk a little bit about PBPK in these systems, and then a little bit on disease modeling. So polymyxins are and antibiotics that are very effective against a lot of different organisms, but they are one of the most nephrotoxic compounds that are in clinical use with very high rates of nephrotoxicity. So one of the questions we asked is whether we could use our kidney MPS to test the mechanisms and come up with preventative strategies. So I'll kind of shortcut here a little bit. This is RNA-seq data where we either exposed proximal tubules to polymyxin B or to just a vehicle. And you can see basically here, so polymyxin are the triangles and the circles are the controls. And you can see I could draw on PC1, principal component 1, a line here that 100% separates polymyxin exposure from no polymyxin exposure. And different donors respond differently. And again, you can look at human heterogeneity, but you get a consistent pattern from polymyxin exposure as you see on this heat map here. And what's very interesting is that we found that the cholesterol biosynthetic pathway was tremendously upregulated after polymyxin B exposure, like 30 of 50 genes in that pathway were all upregulated. Whereas if we use another nephrotoxin, cadmium chloride, there's no upregulation of the cholesterol biosynthetic pathway. On the other hand, NRF2, which is kind of an antioxidant pathway, and that's been druggable, is not upregulated with polymyxin B, but is after cadmium chloride exposure. Now, in both these cases, clinically, you would see acute kidney injury, you would see KIM1 go up, NGAL go up, and clinically, you wouldn't be able to distinguish the mechanism of injury here being different between these two nephrotoxins. So polymyxins are known to be polycationic, and that's thought to be the more polycationic they are, the more nephrotoxic they are. So there was a very small company called Northern Antibiotics coming out of Finland that was making less polycationic structural analogs that still had antibiotic efficacy. So we tested them in this system. And here you're looking at Kim1. So here is polymyxin B, and you see, yes, Kim1 goes up, whereas these less polycationic derivatives of polymyxin B don't really upregulate Kim1 here. And the same for hemoxygenase 1. These compounds don't upregulate hemoxygenase 1 the way that polymyxin B does. And then we showed that these the cationic component integrates into the cell membrane and disrupts the cell membrane. So it makes perfect sense that cholesterol, genes that synthesize cholesterol would be upregulated because you need the cholesterol to repair the cell membranes after they're injured. So we could establish a specific mechanism of injury in this chip system. And then very interestingly, we said, well, that'll be interesting since these cholesterol synthetic genes are upregulated, maybe cholesterol could be a marker in the effluent of this particular type of nephrotoxicity, and it turned out to be true. So with polymyxin B exposure, cholesterol is upregulated, just measuring cholesterol concentration in the fluid that comes out of the chip, whereas after cadmium chloride, it's not. So again, you can see how not only can you test for safety compounds, but you can understand mechanistically nephrotoxicity, I think, a lot more in some of these chip experiments than you necessarily can with animal models. So with that, I'll switch gears a little bit to another question about utility of these chips. Can they be used for modeling PBPK? And in particular, could we were challenged to say, could we make an in vitro to in vivo scaling model for human drug clearance? Of the top 200 drugs sold, 30 of them are cleared by basolateral secretion in the proximal tubule. Now, if a drug is entirely renally cleared by glomerular filtration, that's very easy to model. All you need to know is the molecular weight and the protein binding of the drug. But for drugs that are substrates for all the transporters in the proximal tubule, it's a bit of a black box for PBPK. So we used opioids because there was so much interest in this, and there's a whole series of papers here, about half a dozen papers that we published on this. And I will say this work was led by Nina Iso-Haranen, who is the chair of the Department of Pharmaceutics in the School of Pharmacy at UW, one of our collaborators, and she's a modeler and had just finished a sabbatical at the FDA as well. And it was really her team that did the modeling here. But the long and short of it is that we could model, if you see there is in vivo data, there are published studies about renal clearance of the opioids. And we could compare the in vivo clearance to the MPS clearance that we got, and you can see the ratios here for morphine and morphine 6-guaranide, extremely good. So we literally could make an extremely good in vitro to in vivo scaling model incorporating PBPK into the system. So I do think it's probably the least utilized goal, if you will, for these kinds of microphysiological systems, but I do think they can be used if there's an important question around dosing for in early studies for PBPK. So I mentioned early on that we focused on the proximal tubule. I mentioned early on that the kidney is the most vascular organ. 20-25% of the volume of the kidney are the blood vessels, and the kidney vasculature is poorly understood compared to the vasculature of other organs. And so a young engineer and vascular biologist that we started working with a decade ago learned how to 3D print these vascular networks and make vascular networks that you can actually perfuse with whole blood, and they're non-thrombogenic. They will not clot. So they're really beautifully designed 3D printed vascular networks. And it took us a while, but we learned how to isolate from kidneys endothelial cells grow them out. They're very finicky cells from the kidney. Grow them out, characterize them, put them into these systems. And what you're seeing on the left-hand side here is, again, a photomicrograph of a normal kidney, the cortex. And the blue nuclei are the tubules here. And the two stains, the red and the green, CD31 is endothelial cells, and platelet-derived growth factor receptor beta are these perivascular cells, or pericytes, which make up the capillaries. And the pericytes are always at the inflection points where they can control the hemodynamics of the microcirculation. And this is our in vitro network that we made on chip with the same cell types. And you can see, again, the pericytes are always at the inflection points where they can control the microcirculation. So we wanted to say, could we model any kidney vascular diseases? Because there really has been very little progress in the vascular diseases in the kidney. And almost every type of progressive chronic kidney disease involves what the pathologists call capillary rarefaction or capillary dropout. So we decided to study cyclosporin first, which is an immunosuppressant still used today as an immunosuppressant for transplantation. And the reason we did this is because the preclinical studies before cyclosporin was FDA approved did not really demonstrate nephrotoxicity. And it's still hard to make a good animal model of cyclosporin-induced nephrotoxicity. But when it was approved in the very early days of transplantation, it was given in super high doses, and it caused a lot of thrombotic microangiopathy and kidney loss, actually, from acute kidney injury in these transplants. So we decided to say, could we make a model, an in vitro model of a kidney-specific thrombotic microangiopathy for the first time? So first thing we wanted to do is have an in vitro target engagement biomarker, and it turns out that there's a transcription factor called NFAT, and there's an isoform of it, NFAT1, that upon VEGF stimulation, vascular endothelial growth factor, it translocates NFAT1 from the cytoplasm into the nucleus. So you can see this here, how all the immunofluorescence moves to the nucleus in the center. And it turns out these kidney endothelial cells are extremely VEGF dependent, more so than almost any other endothelial cell. And it turned out that cyclosporin blocks that translocation of NFAT1. And it has turned out really that there's kind of a yin and a yang between VEGF and cyclosporin A as far as the kidney microvasculature. So then we perfused our vascular networks with cyclosporin, and we literally made a thrombotic angiopathy model in vitro. So again, we can perfuse these networks with whole blood. Here we use labeled platelets in green. This is cut open now, the vessel, so you're looking at the luminal surface of the vessel after an hour exposure, and you see these deformed erythrocytes adhering to the lumen and the platelets adhering to the lumen, and their degradation of the fenestrae and translocation of the erythrocytes into what would be the interstitium of the kidney, where they would be highly inflammatory. So we literally made a model, and then we did a kinase regression with 28 different kinase inhibitors and an AI model, and identified kinases that could rescue against vascular injury from cyclosporin, kinase inhibitors, I should say. We then made, I didn't put the slides in, but we recently made another, a shikatoxin model. That's the so-called jack-in-the-box. So when people get exposed to E. coli that has this toxin, they get kidney failure. It's untreatable right now. And we also made a model that works and did a kinase regression and came up with different kinases. So again, the clinical phenotype might be the same, a thrombotic microangiopathy, but the mechanisms, the intracellular mechanisms are different and can be worked on through some of these chips. So, you know, the other component of the kidney is the glomerular filtration barrier. And I will say there have been a number of papers. None of them are super convincing. We've worked on this for 10 years. This is the best. We published this last year. We have for a model where we can use a type of 3D printing that's called subtractive manufacturing to mimic the contours of the glomerulus and create a vascular network. But we haven't recapitulated the glomerular filtration barrier, and I'm not convinced that any of the publications that have worked on this yet have really succeeded. It's a real challenge because the podocyte is such a complicated cell type to get the podocyte to have the right phenotype in vivo. So I'm going to switch gears for the last 10 minutes or so and talk about kidney organoids. Most of the work that we and others have done on the proximal tubule either use primary cells. We get ours either from discarded kidneys from the organ bank that aren't used for transplantation, or from people undergoing nephrectomy, an operation to remove a kidney because they have a circumscribed cancer and the rest of the kidney is normal with consent. And those are good, but you don't get to really manipulate the genome very easily. People are also using immortalized cell lines, and they have their advantages, but they have some disadvantages as well. So the third potential source of cells is inducible pluripotent stem cells that can be made into kidney organoids. And in our group, this work is led by Bino Friedman here, who is one of the pioneers in developing kidney organoids. And you can see here's a human kidney, the major structures in the cortex being the glomeruli, and then the proximal tubules, and then the distal tubules. And this is what an organoid looks like. And it has the same sort of overall basic architecture where the podocytes are on the rim of the cortex, and then the proximal tubule cells are just inside that, and the distal tubule cells are in the center, much like a human kidney. So the overall architecture of these kidney organoids made from stem cells looks like a human kidney, but it's immature. And it doesn't fully replicate the structure, and the cells themselves are more like fetal cells than like adult cells in terms of their functionality. And that's probably the biggest limitation for organoids for disease modeling right now in the kidney, but also elsewhere. Amazing to me is that, you know, healthy people's urine has living cells in it that are sloughed off from the kidney. We can now take a urine sample, de-differentiate that into stem cells, and make a kidney organoid. So it's a little like science fiction, but you can take a urine sample and make a kidney organoid that represents that person's genetic background. And we now have a protocol where people with various kidney diseases around the world can send us urine samples and we can make kidney organoids. And these, what I would say is where these have really shined so far is because of the possible using CRISPR-Cas9 and other genome editing techniques now for genetic diseases. These organoids are can, if you have a good phenotype of the disease, they can be extremely effective. And Bino really led the way for this back in 2015 when it combined stem cell technology, making kidney organoids, and I think was the first person in the world to then use CRISPR-Cas9 to knock out these genes called polycystin 1 and polycystin 2 that cause autosomal dominant polycystic kidney disease, which is still the most common genetic cause of kidney failure. And although there is an FDA-approved treatment, it's not super effective, and the whole world is kind of looking for new therapeutics. And these genes were cloned 25 years ago. PKD1 looks like an orphan G protein coupled receptor, PKD2 or polycystin 2, like a cationic channel, and it's known that they complex together. But 25 years later, their functionality is not that clear, and they tend to go into the cilia. So this was the original work. They're showing that with CRISPR, you can knock out either PKD1, PKD2, and then we wanted to be able to make that into a screen, into a relatively high throughput screen. So we had to increase the cystogenesis rate. So remarkably, when you knock these out in vitro, the organoids form cysts. And Nellie Cruz, working in the lab, figured out a way to get the cystogenesis rate only if you knock out the genes up to 70–80%. So that's certainly high enough for a high throughput screen. And hopefully this movie will play and you can see it. But this is time-lapse photography. Right here, you'll see over a period of weeks, a cyst actually form in an organoid and pop out. So we can see biophysically how a human cyst, so this is gene specific, because you don't get cysts if you don't knock out the polycystin genes using human cells. We have a beautiful phenotype for disease modeling in vitro. And then we did develop, using robotics, a higher throughput screen and came up with pathways that had, after 25 years, had not previously been identified relating to the cytoskeleton, actin-myosin pathways, as a new potential pathway for treatment in polycystic kidney disease. And we're exploring that now in animal models. Now, another natural thing to do that occurred to us and to others is to take these organoids, and they could be, in our case, we're talking about kidney organoids, and put them into chips. So if you combine flow, dynamic shear stress flow, with the cellular architecture of the organoids, can you get further maturation, differentiation, and functionality? And this is still unpublished data. But yes, when we put our PKD organoids under flow in BD chips, they oscillate. You can actually watch over time the cyst size change. It grows, it shrinks, it grows, it sinks. But overall, it grows much faster under flow. And we've also seen maturation and changes in the functionality of the cells in these organoids under flow, as have others as well. So these technologies are now being combined, organoid technology and chip technology. And this is, I put this in because I think this shows ground truth fidelity of an organoid model. Over on the left here is one of our grad students getting her hazmat outfit on as she's going to go to a BSL-3 hood and inject live SARS-CoV-2 virus into organoids. It's known that in the early days of SARS-CoV-2, there were very high rates of acute kidney injury, and the main receptor for SARS-CoV-2, ACE2, is highly expressed in the kidney. The kidney, the gut, and the lung are really where the receptor is expressed, these large epithelial surfaces. And so sure enough, it turns out you can transfect or infect kidney organoids with SARS-CoV-2. And it mostly affects the proximal tubule cells. There is a therapeutic being developed from the Institute of Protein Design at UW, a nano particle-based therapeutic that's now moving into clinical trials. And we tested this system and found, yes, we could prevent, in a dose-dependent way, we could prevent plaque-forming units. But this was the part that to me was particularly important. So one of our other investigators at UW in the Kidney Research Institute had a grant from the CDC early on to study SARS-CoV-2 in people in the ICU with respiratory failure, and he collected urine samples from about 100 people on ventilators with SARS-CoV-2 in the early days and compared them, the urine samples, to people who were also in the ICU on ventilators, either from H. flu or from bacterial pneumonia, and used the somologic approach, and came up with 9 proteins that were differentially expressed in the urine of patients who had respiratory failure and critical illness from SARS-CoV-2, as opposed to other bacterial or viral causes. And in our kidney organoids, after we transfected them with SARS-CoV-2, 8 of those 9 proteins, the genes were transcriptionally upregulated. So fidelity between what we see in our in vitro system and what's seen in vivo as well. And I mentioned that people are now combining, just as we are, flow with kidney organoids. This is nice work from Jennifer Lewis again and Ryuji Morizane in Boston. Again, putting vascularized kidney organoids on chips and showing that they get real maturation and increase in the vascularization, or at least the number of vascular cells in the organoids, which you can see here. Here's you can see static and then low flow and then high flow, and yellow here are the vessels, and you can see much more of a vascular network forming and a more mature vascular network forming when you use flow. So I was asked to sort of summarize sort of where I think this field is right now. And it's been 10 years literally since I think we were the first group funded to make a kidney on a chip through the NCATS program, literally 2012, so 10 years ago. So what I would say is for the proximal tubule chips are very successful. ours, others, as I mentioned, there's 6 or 7 at least different commercially available chips. I personally don't think the chip is as important as the cell source. But if you want, I think they're proven to have utility in safety. They can be used for disease modeling, and they can be used for physiologically based pharmacokinetic modeling of drugs as well. So those, I think, are pretty mature at this point in time, and their utility has been demonstrated by multiple groups, I think, in both industry and in academia. The critical variable is really the cell source, I think, whether you're using, and each have their advantages and their disadvantages. So it's really context of use and what you're trying to accomplish. Primary cells, immortalized cells, iPS-derived cells. Kidney organoids right now, very dynamic. It's like every month there's another paper published, and it's a maturing technology. It's probably a pun there because one of the big challenges is the cells are immature. But there's a lot of problems still. And lots of groups are working on this now. And again, industry is adopting these, but with a lot of appropriate, in my mind, skepticism at this point in time, but trying to learn more, as are we in the academic world. So the challenges are really about maturity of the cells. They don't really vascularize per se. They have endothelial cells, but the vessels don't connect, and they don't, there's no blood in there. What people are learning is that if you do take kidney organoids and transplant them under the capsule of a mouse kidney, the mouse vessels do grow into the organoid, and they actually do vascularize the organoid. So from a regenerative medicine standpoint, there's a lot of promise here. And the physiology is not integrated in the organoids because, yes, you have all the cell types, but they're not interconnected. There's no fluidics. In the same way, there's no luminofluidics the way you would have the proximal tubule, all the different segments of the nephron tubular epithelia interconnected with each other. So they don't have the same physiology as you can make, let's say, with chips. I think the kidney organoids right now are most useful for genetic diseases using genome editing techniques if you've got a phenotype on chip. Polycystic kidney disease is one. There are other genetic diseases we're modeling, other people are modeling now as well. The key is whether you have a good phenotype or not. And at least to my mind, the glomerular models, including ours, and we have struggled with this for 10 years, are problematic because the podocyte is such a complex, unique cell. Nobody has been able to really get a podocyte to look like a podocyte in vitro. It's a terminally differentiated cell in vivo with these 3 different levels of foot processes that adhere to a basement membrane and form a filtration barrier, and that really hasn't been very well accomplished. And I think that's the last slide, except for thanking all the people who are here, all of our funders, and also the people who do all the work. Ying Zheng, as I mentioned, is an engineer and a vascular biologist. Ed Kelly is a toxicologist. Friedman, stem cell biologist and kidney modeler. And Kathy Young is a PharmD PhD who does all our pharmacokinetic type work. So thanks for your attention. I think I saved some time for questions.
You sure did. And thank you so much, Jonathan. That was a really fascinating talk that gave a really great overview. I do have a question already. One person asks if there have been any efforts to date to add immune components.
Yeah, good question. Yes, there have been, and it's a very important question, obviously. It's challenging because you can embed cells in the interstitium. We've done that in these chips. But the question is, which immune cells do you put in there? There is a lot of work going on through AMP, the Accelerated Medicines Project, for example, where kidney biopsy, that's unrelated to chip work, but that's also kind of a public-private consortium where industry is involved. And they've studied, for example, lupus nephritis, which is an immune kidney disease. The challenge is there are resident immune cells in the kidney, different types, in different parts of the kidney. There are also infiltrating immune cells in any kind of either autoimmune disease or inflammatory disease that involves the kidney. So, and what AMP is doing now through single-cell RNA-seq is they are describing under different conditions, particularly with lupus, these are human biopsies, which immune cells are in the kidney at what point in time. We're also doing that. I'm the chair of a steering committee for an NIH-funded study called the Kidney Precision Medicine Project, where we're also interrogating human kidney biopsies in diseases like diabetic kidney disease and acute kidney injury. And we're starting to phenotype all of the different immune cells that are in the kidney. But it's very complicated. So it's one thing to say, okay, I'm going to take some lymphocytes and I'm going to put them into my model and see what happens. But T cells, B cells, macrophages, we are modeling now. We are working on in vitro a model of lupus nephritis. And we are working with a company on that, as well as funded by Department of Defense for that. But knowing which immune cells to put into your model to replicate the process, you know, some immune cells come into the kidney, or if they're probably resident macrophages, are reparative in some cases. For example, after acute kidney injury, they can be reparative, but they can also be harmful. So it's one thing to say, oh, I'm going to throw some cells into my system, but I would say it's at a very, from our perspective, and I think others, it's really early for that.
Yeah, that's a great response. Thank you. I am going to quickly put in the chat the link for the Kidney MPS workshop, just in case anybody hasn't seen that, so they can register. If you do have any other questions, feel free to put them in the Q&A or the chat. I will see them. One question that I have, and kind of related to kind of the complexity of this, is do you have an understanding or an idea of how these kidney MPS can work in collaboration and coordination with models? Like when you're thinking about like large scale research, are there certain areas where you think, you know, should definitely be done more in kidney MPS, some more in animal models? Do you have a sense of that?
Yeah, so it's variable. First of all, they tend to be high content, low throughput systems overall. So they're not going to, you know, people, you know, I don't know how many different companies have come up to us and said, can you, like, you know, run a screen of 200,000 compounds or 2 million compounds? And the answer is no to that. That's not what they're going to be good for, that kind of thing. On the other hand, I remember talking to a safety officer in a large pharma who said, you know, we don't really need your MPSs when everything's going well. But if we're in the middle of a phase 3 clinical trial and we encounter an unexpected nephrotoxicity signal and we have to stop our trial, we have 100 plus sites around the world and we have to stop our trial, we will pour money into whatever can give us an answer. So, you know, you have different attitudes for different purposes. For one of the other questions industry always asks, and we're actually funded to look at this now, is if I see a signal in a human MPS, a safety signal, let's say, that I don't see in an animal model, is it due to the fact that these are human cells, or is it due to the species difference? Or conversely, if I see a safety signal in an animal and I don't see it in your MPS, is it because the MPS doesn't pick it up, or is it because you're using human cells? So one of the things that IQ Consortium for MPS has always said, I think correctly, is we should make dog on a chip. We should make all kinds of species on a chip. We are starting to do that. We got an SBIR grant to do that. So I don't know if I'm answering the questions fully, but.
No, that's great. It's a big question. So yeah. I think even those answers are really wonderful.
Yeah, and I will say that they fit into different companies, fit them in different ways. Some are more, you know, there's been healthy skepticism, but also as IQ Consortium has watched the development, they're showing much more utility as time goes by. Some companies, it's more on the safety side, some it's more on the development side, some it's on both.
Yeah, absolutely. That's super helpful. Well, we are just past the hour and I don't see any. Well, okay, I have one question. Do you have time, Jonathan?
I have time.
Okay, then I'll go ahead and ask it. So emerging single cell or single nuke seq has revealed failed repair subtypes of proximal tubule cells that have been observed in 2D cultured cell in vitro. Have you looked into the single cell analysis to characterize the subtypes within MPS?
Starting to. Yeah, and there is this whole senolytics idea that there is a phenotype of a failed repair cell that actually there's now attempts at therapeutics. If you eliminate those cells, then the kidney and other organs as well. Senolytics are now being looked at for aging by a lot of companies, reverse aging, et cetera. So yes, the single cell technologies we're starting to apply RNA-seq and ATAC-seq to the chips. What we haven't done yet is the spatial resolution, also at single cell level for transcripts and for proteins, the technologies that are emerging. So we're just starting to incorporate that into sort of the workflow. I will say it's expensive when you're in academia. Single cell work is expensive to do, so you have to design your experiments carefully.
Absolutely. Well, thank you for all those answers. I wanted to just give you the opportunity. Is there any final words that you want to give to the presenters today or the attendees today or anybody watching the recording?
No, nothing in particular.
Awesome. Well, you gave a really wonderful talk, a really great overview. Really thank you for your time. And everybody have a great rest of your day. And thank you again. Okay, bye everyone.